Okay, now we are in streaming, and I'm presenting here this presentation.
So my presentation is biological properties of the South Adriatic. I represent our data. Our data with, in combination with some data with other people here down the list. And this data is collected within our project ADMET plans financed by the Science Foundation. And part of the project is to investigate the evolution of this phytoplankton bloom at the open sea under the influence of these interconnected events. So our station is here in the South Adriatic. It's 1,200 meters deep, and this is the direction of the Bura or Bora wind, which blows from the to the open sea. Now we sampled in January 2017, on 20th of January, and already you can see this is chlorophyll data collected with this chlorophyll sensor of wet labs. And you can see that the chlorophyll values are really small, and the chlorophyll is extending up to 450 meters here, even at the end of January. And here on this graph, you can see this like integrative value of this on per meter squared. So on 8th of February, there was even deeper convection of 650 meters, and it is 19 days from the first sampling. So you can see the chlorophyll elevated a bit, but not too much. But this convection even went deeper, so there were some convective events between these samplings. So the next sampling was on 16th of February. So you can see that this phytoplankton which sunk, it was 8 days ago, degraded here, so it's at 0. And here this like surface chlorophyll maximum started to form. Next sampling was in March, at the beginning of March, and you can see now that this chlorophyll peak is forming. It was 15 days after, and then the big peak was on 20th of March. And as Laura said, you can see that there are like 2 months lag between this start of these convections and the peak of the chlorophyll. So we think also that the development of this peak of the chlorophyll probably depends on how many of these convective events there are and how strong the Bura blows. And if this Bura will also blow with high intensity at the end of February and March, then the peak will probably shift towards April. So these Bura events probably influence the evolution of these peaks and probably shift it later if there are many of these events. So we also saw on Laura's slides that these Bura events also influence zooplankton in a way that it carries a bit of this zooplankton downwards, but not too much, because the majority of this, at least mesozooplankton, can resist this vertical mixing, and then within 5 days they return to their original position, so this ADCP signal is restored after a couple of days. But the problem with this vertical mixing is that it dilutes this surface chlorophyll, which is available to zooplankton. So if these convective events of high intensity last, they probably cause starving in the zooplankton, or at least lower the abundance of zooplankton. So unfortunately, after this date, there was like some kind of new water coming, and it displaced this water mass. So we couldn't track anymore this evolution of this chlorophyll maximum. And for the rest of the year, what the open sea chlorophyll is usually like this. This is situation in July. You have like small peak downwards, and that's almost to the end of the year. The situation is more or less the same. So we also have this satellite data from long-term satellite data, this Hoff-Müller diagram, and you can see also that the winter is here the main season of production at the open sea. and probably these other seasons are not so productive, even though sometimes in autumn we can see some peaks, but that usually does not correspond in a like huge boom of zooplankton. Regarding zooplankton, the situation is similar. We have this peak in March, and then it declines. So these zooplankton values are really small throughout the summer, and sometimes even you can see these are dead flares. You can even see that this dead flare from 100 to 200 even can be more abundant than this two surface flare. And in the autumn, you can have this like small secondary peak in zooplankton production. So this is one interesting point from this paper we did together, because these ADCP values are high in summer months, and they are high night and day. So it means that there's something standing in this surface layer which is causing this box scatter signal. So we assume that these are probably signal from this gelatinous zooplankton, which is not really studied as it should be studied, because it is hard to sample it. Usually these planktonids which we use are not quantitative for that kind of plankton, and they are big organisms which you miss with net samplings, and they have huge impact for the open sea ecosystem. So we think that probably these big signals in summer when all other net zooplankton is low is probably due to these gelatinous organisms.
Sorry, is it possible to read what you are describing?
So these are on different depths, these backscatter strengths, monthly values through the years. So you can see the highest values are in summer, so these are months, these small dots. So this is 1 year, and this is
maybe the depth. This one, the red one is the
65 meters,
Yeah.
100 meter is that here, and 150 and 200
Yeah, but
meters.
this is what's proportional to what? To the concentration, to the abundance, to the...
No, this is,
It is the backscattering
The
strength. It
backscattering.
is
DCP.
the DCP.
Yes, so is, I mean, just you know that something...
we know that something is there, but that there is a lot of this something than before, when there is practically nothing in the nets. So there's must be something else which we don't sample. So,
okay.
So the only thing which could be, which is stationary, there's no day and night signal. With this, it is just stationary. It could be the only probable thing likely which could be is the sulfs or ctenophores, because
Just a question. Just understand to follow you. This is the signal that you measure in the BOA
Yes,
With the
Vanessa?
with the
Yes.
DCP, with the DCP.
and the other one were the data of your station, okay?
Yes.
the other is on the stage, yes.
Okay.
But that's zooplankton, but because it is in the South Adriatic, we can assume that it is
Yes.
more or less the same, because it is inside this. And it is a constant signal throughout the years. This is like typical. This is not like just. This is like typical zooplankton abundance at the open sea at that time. There's nothing practically during the summer. It is like really low. Even this peak is quite low in comparison with close to the shore.
There's a reason that I'm not so sure that I installed, because we got data during the Project Sesame on the Italian side, and the abundance is very higher. But
Yeah, on Italian, yes, but here on the Croatian side, it's much more oligotrophic.
Okay.
So this is one of the probable species which could cause this signal. This is Salpa maxima. It is big salmon. It is like 10-15 centimeters. It's huge filtration rates, and we see it constantly at the open sea in spring, summer. You can see one or two, but it is hard to quantify them because you see what is on the surface and you don't know what is below. And this species, in literature, there is not a lot of data of it, but it is probably in the first 50 meters of depth. And you can see that it is interesting that in June 2009, when we were sampling, we saw a lot of them, and at the same time, we saw really small amount of copepods. And it is also possible that these salps can put stop to this phytoplankton bloom in the spring. It can happen that they start to appear in spring from April to June, and they can consume a lot of this phytoplankton, and they have really big fecal pellets which sink rapidly, so they can deplete the surface layer from the nutrients and put an end to this phytoplankton, or at least limit it. So this is this situation in June 2009. You can see a lot of them floating around. So each of these squares is one zooid, and they are like big with huge filtration rates. So we estimated about 1.5 per square meter, but what's down there, it's really hard to say. And they are specific in a way that they can also reproduce sexually and asexually by budding. So they can, in short time, have huge changes in population number if the conditions are right.
What is this, a picture?
This is projection from the boat, surface.
From the boat.
Yes, surface from the boat to the surface of the sea. So you can see a lot of them floating around at the open sea. So this was like huge boom. And they had a huge impact for the zooplankton community. So the other part of this talk is the influence of this bios system on arrival of these alien species to the of zooplankton to the Adriatic Sea. So according to this bios theory, there is in Ionian Sea, there is a jar which changes its direction on the Cardial scale, and in its cyclonic regime, it draws more water from Eastern Mediterranean, and anticyclonic, it draws more from the Western and Atlantic water in the Adriatic Sea, and it also brings more of species from one or other side in the Adriatic and changes the composition of Adriatic zooplankton. So these are the station which we sampled, and our data show that this can be seen as SST anomalies. This is a long-term trend of salinity and temperature from 85 to 2011, and this is average from 200 to 800 meters of this salinity and temperature. So we have here in this cyclonic phase, you have higher salinity and then lower salinity in the anticyclonic phase. And here down there is a list of species which we found in period of 93 to 2011. And these black squares indicate when the species first time appeared in the Adriatic. And this plus means it was in really low number, so we found maybe 1 or 2. And these colors are contribution of each species on its respective group. So, for example, if you have this Bruxia rostrata, then this color here, that there's no color because we found one, but the color would indicate that it, for example, here is about 30% in the salps. It contributed during that year. So, these species, arrival of these species, we can divide in 3 groups. The first was in this anticyclonic phase, cyclonic, and then again anticyclonic phase. And if we divide them by their temperature preference, you can see that most of the species which appeared are like warm temperate species, and few of them are cold temperate species. And these cold temperate species, they all came here during this anticyclonic period, when this warm temperate species, they could come even in one or the other period. So the other thing is that these species which entered the Adriatic, and it seems they are now established here, their abundance or contribution within their respective group depends on this biocycle. So this warm temperate species, even though it entered during this anticyclonic cycle, its abundance increases during this cyclonic, because it's probably what the conditions for this species are better in this part of the cycle than in another. So in this cold temperate species, you can see how they decline during this cyclonic period, and then they start to rise again. And So the next thing we are now doing is we are trying to do this genetic approach, because the problem with these species, we can assume where they came from based on the biocycle, but we are not sure. So one approach we can take is to use genetics to see if there are any differences in these eastern and western populations, or even from the Red Sea, to see where they could have come from. And in this 2014, we found a new species and an alien species, new species totally for the science, and we sequenced it, and we saw that there is 1.5% distance in this 18S RNA, which means it is a different species than one. In this genus, there is practically only one other. So, but until we found this one, it was just one species. Now we found the second in the same genus. So we thought, okay, this is a new species and an alien species which entered the Adriatic, and we saw that there is difference even genetics, but this is difference in morphology. This is the already known species, is Bruxia rostrata. This is our species, is Bruxia lacrome. So you can see there are like huge differences. This one has like 4 muscles. This one has 3 muscles. So that's huge for us. And then this muscle here branches in 2, and they are here connected together all the time, and then branches each separately. And this is also subspecies, so it has 2 generations. This is like solitary form, which reproduces asexually, and this is aggregate form, which reproduces sexually. So they are completely different. And then we tried to compare these sexual forms. So this is already known species. This is our new species, which is also, you can see like there's a huge difference. There are like 3 muscle bands coming here, and here are only 2, and the third is here. And also in this process, there are like 2 muscle bands, and here are 3 muscle bands. And this one here connects behind to this one. So that's like huge difference for this group. Yeah. But we found, after we published this paper, a drawing from Fedele from Napoli in 1926. He drew the same thing. And in his paper, he said that he at first thought it was a new species, but then when he saw the identification piece to this group, he concluded that it is not the same new species, it is the same, this Bruxia rostrata, but the keys were really bad. So these are the keys he had to deal with. So it's not to blame him that he thought that this was not a new species. But it is interesting that this species was recorded in 1926, and then for 80 years nothing. And if we didn't find this drawing by Fedele, we would think that it is new Mediterranean species. And in fact, it is all along here, but it is probably not sampled because there are not many experts in this group. So that's also one big problem, because you need to assess really accurately what is a new species and what is just new to you because it is undersampled group. And the next thing we did this Thalia orientalis, which we found in 2008 for the first time. It had like 3 possible routes to enter the Adriatic. One route was Atlantic, the other route was Eastern Atlantic, then Eastern Mediterranean, then Adriatic, and the other route was from the Red Sea because it is cosmopolitan, warm-temperate species. So we did genetics of this Adriatic form, and we did genetics. We were able to get samples from Pacific, and then we saw that there are like 2% difference. So it means that there are completely different species. So it means that this species came to us from this part or this part, but not from the Red Sea. And this 2% difference and this morphological difference, which doesn't look too much, but they have this 2% difference in this gene is, I think we are closer to cow than they are together in genetic distance. So it's like huge distance, but morphology is really the same. You can see that this part is a bit different and they are a bit more longer. So it is also, the next important tool is this genetics, so that we are able to see where these species come from. Are they new or they are not new, or they came from Red Sea, maybe through Suez, or they came towards the Atlantic. And now it seems that this species which we have was thought to be this Italia-Messico, but it's in fact new species. And it seems that this Atlantic has its own form of this Thalia orientalis. So that's almost it. We have this.
So the conclusion is where this comes from?
This is probably Atlantic. It is our
Okay.
local, but because of this very similar species, this was originally described. This species was originally described as Pacific, and it is not Pacific. We see by the Pacific, or we see by the genetics. So it's a new species, in fact. But they thought for years that it is the same because you couldn't have a like objective way of telling. The differences are really subtle, so you don't know if it is natural variability or it is a real difference, and it's in fact a different species. So you have genetics to help you inside. So we also did genetics of some other species which are alien to Adriatic. For example, this Pelagodia longicirata is in fact a complex of cryptic species. They all look the same, but there are like 5 or 6 of these, and it is really hard to tell them apart just by looking at them. So there is like Google for sequences, there is a gene bank, so we compared our sequence to this gene bank and we find 5 or 6 sequences, but none was identical to this one. So it is also some kind of new species, and it is also a big problem, this cryptic diversity. You have these species which look alike, but they are not the same species. It is hard to tell them apart, and if you don't do genetics of your population, you can think that nothing changed, and you have, in fact, like this cryptic invasion. You have another species which came, which is not local. And we also did this genetics of Mugia atlantica, which we know that came from Atlantic, and this Atlantic and Adriatic population were identical in their genetic makeup. It means that it probably came in some number, so it was genetically preserved. It didn't change its genetic makeup, because if you have original population and small part of this population moves to another place, if this population is too small, then they are usually not exact genetic replica of the original population. And the third thing we done is this Salpa maxima, this big salp of the open sea. We sequenced that one, and it showed that the sequence was identical to Pacific and not to Atlantic. This species. So it might be that in the Adriatic we have now a mix of these Salpa maximus, one which are like local and other which came through Suez, but this needs a bit of more work to be sure. But it is interesting, it is more similar, it is identical to this Pacific and not to Atlantic.
How many specimen did you analyze?
One
Just
for
one.
now, yeah.
Maybe more specimen are
Yes,
necessary.
this is preliminary.
Check
Yeah.
as strong.
Yeah, but this is just preliminary, but so far it seems that this Atlantic basin and Pacific are more or less separated. At least regarding of these warm temperature species, it seems that it's not easy for them to cross from one side to another. But we now have this Suez Canal, but so far we cannot tell for certain, at least for these species which we have in the Adriatic, that any species came from that route.
But don't you think that is a problem also of number of sequences? Because
That's also a possibility.
At the end, for the moment, genetics is just a play of statistics.
Well, for some things, for
More or
some,
less, because they compare sequences, but if your reference sequence is based only on one individual,
yeah, but it is possible. For example, one possibility is that the populations are the same, Pacific and Atlantic, and that we have this mixture there in Atlantic and Pacific. So we have everywhere the same things.
but I'm promoting the variability in the population.
Well, because this 18S gene, which is really conserved. Even one mutation here usually points to a new species. So it is, at this point, for me, it is more likely that there are like 2 cryptic species of Salpa maxima than there are like variability in this gene. In some other genes, there can be a lot of mutations.
And the question about Atlantica, what do you think? Can we still consider Atlantica as an alien species for the Adriatic?
Well, no, now it seems that it is established. we even here, where is it?
Because this first record dates back
It's-
to
95. Now it is well established. When it came for the first time, it made a huge population in North Adriatic, and then the number of small cockroaches declined. But now it is
Okay, now you know.
here, so
Yeah.
we have to get used to it. But it seems that its number drops in cyclonic phase and it rises now. In this cyclonic, we also find really low numbers of this Atlantic. It was there, but in really low.
Okay,
So that's it. Thank you for attention.
thank you very much. Quick questions?
Very
Okay.
quick. Do you have elements to distinguish whether these species came by natural waterways or by
Yeah.
marine traffic?
That was all I wanted to ask this one.
No, but I think my personal belief is gelatinous things, most of them are too fragile to, I think, be pumped in and out. They are really fragile, so they will break up.
Nemiopsis maybe, for example, are what arrive with the
Yes.
maritime transport.
That's
Because Nemiopsis is considered an alien because according to our actual marine strategy, the alien species are just the species that arrive by human
yes.
transport. So species that enter from the Atlantic through Gibraltar are not considered alien species according to this directive, while people, species transported by human in ballast water, or for example, aquarium release, aquaculture release, or through the Suez Channel, which is a human channel.
non-indigenous species, the terms that are
For example, we were asked by the reviewers to use it. Okay. Yeah.
No, because
Se no le non
education,
Yes.
but non-indigenous.
Yes, it's more correct. Yes.
Okay, I'll give the floor to Stefano.
Okay, thank you, Vanessa, for inviting
I
us.
Yes, I was to say is a half an hour presentation since we have the problem with it. So I will give her, as I said,
I will try to do as
25
fast as
and
I
then
can.
we'll ring, I'm sorry.
We can merge this presentation.
I'm sorry, just because I don't want to make you lose the train.
Okay, I will try to talk to you about the particle transfer processes that we are looking, when the other see in which way we collect the particle fluxes and where are located our studies. And then I will present some of the main results that we obtained from past and ongoing experiments. And then I will discuss about the factor that influence the particle fluxes and possible to detect some long-term trend on the particle fluxes. So what we are looking for? Basically, with the sediment traps, we collect vertical fluxes of particles that are connected to the biological pump, so phytoplankton uptake, and then export of organic carbon to the bottom. But also we look at lateral direction or resuspension that occurs transporting sediments from the shelf to the different part of the basin. I can just skip this slide because we already discussed it, was already presented the circulation, the general characteristics of the Adriatic Sea. And so these yellow dots are all the sites of the experiments that we have conducted in the last 25 years almost. Everything started in 94-95 during a European project in which we were comparing different sites, all of them at the same depth, 1,000 meters in different environments of the Mediterranean Sea. And then we continue to study the center of the basin to look at the vertical transport of the whole. And then we focused also with different experiments on the western margin because in time to look better to the lateral advection of particles in the basin. This is just an example of how is made our moorings. Usually we have a sediment trap that is located 35 meters above the bottom. And then just below that we have a CT recorder. And we try to measure the field of the current in the last part of the water column, in the last 100 meters. When we do these studies in the center of the basin, there is another additional sediment trap that is located immediately below the photic zone, more or less about 150 meters below the surface. Which kind of measure we do? Each sediment trap has a carousel, so we can set up a time interval of sampling, and we measure mostly the mass flux, and then we do the composition of the material that we collect in terms of organic carbon, both biological particles and lithographical particles. This is one of the first results that we obtained in one of the past experiments. That was the demonstration that the open ocean convection acted as a controlling mechanism for the spring phytoplankton bloom. As you can see from the upper, from this upper layer, the water column presents the stratification situation with nutrient distribution only in the lower part of the water column, but after a period of open ocean convection in the center of the basin, nutrients were injected in the surface layer, and so started an increase of the primary production, in case that was detected by remote sensitive chlorophyll A. And our results that are the blue curve represented here. These are presented as a time series of organic carbon flux, showing that after the period in which open ocean convection and inaction of nutrient in the upper layer occurred, the organic carbon flux remained elevated for a long period. Then here's, this is another experiment that was conducted. Another experiment that was conducted in the western side of the basin, taking more account of the bottom morphology. That is present a structure that is called the Barry Canyon, that is are two canyons, a system made of two canyons that favor the transport of the sediments down in the deepest part of the basin. The experiment was conducted using three different moorings. Two of them were deployed within the two main branches of the canyon, and another one was just set as a reference on the open slope, not affected from this constraint of the morphology. And our result showed that this is 2005, as you have seen before. He was presenting a sketch with the timing of the variation in time of heat loss. So probably also year 2005 was a good year for the right year for a lot of formation of northern Adriatic dense water that was detected from our instruments. That was detected from our instrument, showing that there was an increase of current speed, and the increase of current speed was more strong in the southern branch of the canyon that has more confinement of the current, and this increase of the bottom current was related occurred together with decrease of water temperature. So that demonstrates that there is a strong interaction with the morphology, and that these canyons are very active in transferring sediments towards the bottom, toward the deep basin. In fact, if we look at the total mass fluxes that we have recorded in the 3 station, the total mass is presented the temporal variability that occurred simultaneously at the 3 stations, with the peaks in the early spring, in late spring, and then a general decrease, but show a clear difference in terms of intensity, because the maximum flux was found in the canyon, in the southern branch of the canyon. That was almost the double of what was measured in this branch of the canyon, and that in the open slope was also visible a signal of increase in transport of material, but with less intensity respect to the other 2 stations. We have used a lot the geochemical characterization of particulate organic carbon, trying to understand the provenance of these particles. For example, on the samples of the experiment that I just showed you, the trap sample, these are the green and the blue circle present here. They were divided into groups. One group is the one with the low fluxes, and the other one, the blue group, is the group with the period of high fluxes. So what we have related to Northern Adriatic dense water cascading, and we have seen that the composition of the organic matter of this sample, the one transported from the cascading, was very similar to the composition of the surface sediments collected on the shelf just in front of the Valley Canyon. And was also compared the difference, the composition of the sediments, for example, carried from a river like the Po River, that they were completely different from this one. So, whilst the sample collected during the low flux period present a composition of the organic matter more similar to the phytoplankton material. During another experiment conducted in the center of the Southern Adriatic Basin during year 2007-2008, just to show you, we were comparing the organic matter composition in terms of delta carbon 13 and the nitrogen carbon ratio, showing that the sample collected with the bottom, with the upper trap, the one just below the photic zone, is a organic carbon composition very similar to phytoplankton composition, whilst the sample collected in the bottom traps, the one close to the bottom, present the composition more similar or very similar to the sample collected in the Valley Canyon. Then another experiment just before was explained that during February 2012 there was a very strong cold outbreak that occupied all the northern hemisphere or the northern Europe, and these are the consequences was shown before, so I can just go very quickly. That just to reinforce also during those days also the water of the Venice Lagoon became frozen. That's something very, very rare. But it's important to confirm that there was an extreme cooling down of the superficial water, as is shown here from the satellite images of water temperature, that caused a lot of thank you. That caused a lot of the production of a huge amount of Northern Adriatic dense water, but that water was also particularly dense because it was from occurred also a period of low discharge from the main input of fresh water of the Po River. So these cold outbreaks gave us the occasion of to design a rapid response experiment, trying to follow the spreading all along the Atlantic Sea of this Northern Adriatic dense water, and that was approached with a multidisciplinary experiment. so involving modeling to trying to forecast the timing, the arrival time in the different zone, to follow the pathway. We also increased the deployment of the mooring. Two of them were already there working, and we added 3 new stations that were positioned taking account of the bottom morphology, because the morphology of the western and southern Adriatic margin present a very detailed, a very characteristic morphology with a lot of signature that can be the evidence for a particular pathway of the bottom water. And also we're conducting several rapid response hydrological cruises trying to follow directly the signal of the water coming out. So this could be one of the possible paths of this Northern Adriatic dense water. So the idea was to study the Bari Canyon and the Bari Canyon system, since we have already seen that that place was one. It was important for cascading transfer, and also in the open slope just in front of the Gargano promontory, and also another possible path of arrival of Northern Adriatic dense water after its interaction with the individual Adriatic depression was to deploy a mooring here, just off of the Pelagosa seal, but this was more complicated because this station is located in the Croatian water, so it was not possible in such a short time to have the permission to do the deployment. So we came out with 5 different stations. As I told you, the 2 in the Bari Canyon, 1 in the open slope, that is more or less the station off of the Gargano promontory, 1 in another sediment mud waves area just off, that is a kind of a continuation of the Bari Canyon, and another point just in a moat that is surrounding the down seamount that is structured on the bottom of the basin. So here I'm showing the results that we obtained in terms of mass fluxes. These are the blue, the brown plots, and variation of temperature and current speed in these 5 stations. What we have detected is the cascading occur not in just a single event or continuous event, but these are almost 4 months of measurement. The cascading occurs in multiple pulses that has separate frequencies, and these pulses of these peaks of negative temperature were related to increase of bottom speeds, bottom current speed, and there was a simultaneous increase of bottom total mass fluxes close to the bottom, but the intensity of this peak is not directly related to the distance, for example, of from each station to the dense water formation area, but is more related to the impact that has the seafloor morphology on because, for example, fluxes were either mooring F and mooring EE are joined together, we can say, and are the water that crosses to F then arrives to EE, to station EE. But, for example, fluxes are more either in station EE because there the morphological confinement is stronger. And the same occurred here in station BB and CC, where the canyon is more effective is the southern branch, and the mooring DD that is outside from the canyon on the possible pathway, still you can see an increase of the fluxes, but with a lower value respect to this station. As I told you, two of these stations were already working on the shelf. These were the station BB in one of the branches of the canyon, and the other one just south of it. And as you can see, here we have the blue curve present the potential temperature trend in the station, and with the arrival of the dense water cascading, temperature was a sharp decrease of about 0.8, 0.6 degrees. And the same was detected more or less at the same time in mooring DD. And these different behavior of temperature occurred simultaneously with increase of in current speed. The same occurred also for mass flux. When started the increase of current speed, there was an increase of mass flux, and even if the long-term trend, I mean, the year before presented always the same seasonal variation with higher fluxes during springtime. Here, again, I want to show you just how it was important, just looking at the period of sampling, the following period of sampling, always in the 5 mooring station, where you can see that this is the highlighted one is the period of, we call it the cascading, with the 4th month, and then after that there was temperature starts to increase a little bit, and there was never very few negative peaks, and these negative peaks was not so evident, and this occurred almost everywhere on the station. And also in terms of the mass fluxes were higher during this period, and then there was a general reduction in all of the station. Another result that came out from this experiment is that usually the literatures say that the arrival in the southern Adriatic of the first signal of dense water generation in the northern part should occur after 1 or 2 months. But we were able to detect the arrival of the dense water, of the cold water, early, just maybe 10 days after the formation, the starting of the formation in the northern Adriatic, or in the northern Adriatic, or in one of the sites just offshore of the Croatian coast. And also the site B, so it's a very good site as a proxy for the shelf dense water arrival in the area. This is the complete time series that we have in that station, starting from January 2010 to January, more or less 8 years of measurement. As you can see, these peaks, negative peaks, is the winter 2012 episode, and then there was some episodes, but very limited in time, and then probably there was another quite good episode that occurred last winter, this winter in January 2018. Here we have another experiment going on together with we take the advantage of the presence of A2 M3 observatory, where we have the 2 sailing clubs, as was indicated this morning. And here we have we are measuring particle fluxes. The particle fluxes continue to present a seasonal and terminal variability. For example, usually there is a increase of the fluxes during the spring, and then a general decrease of the rest of the year, but the intensity in the different years are different from one year to another. And then there is this general trend that shows that the lithogenic and total mass fluxes are higher on the bottom respect to the surface. That is an evidence of lateral transport of the sediment. But what is very important that now the interpretation of the result is in early stage, and it will be very important to have all the registration of the situation of the thermal variation of the thermal situation of the station with with all the instruments that the observatory contains. Another thing that we were looking in this sample is the fluxes of metal, in this case, because we are measuring also contents of some lithogenic elements like aluminum, iron, and manganese, and some anthropogenic elements like cuprum, nickel, and lead. And we have evidenced that there is a common mechanism for all these elements. They come all together, whether if they are of crustal or anthropogenic origin. And we calculate enrichment factor respect to the composition of the local and regional background for the area, and we have seen that cuprum and nickel do not present a high enrichment, whilst there is an indication of some anthropogenic input with lead. And this also, if it's only a moderate input, but it seems that there is a kind of a constant input of lead from to the deep part of the basin. I was also trying to detect if in the experiment we have conducted in the central part of the basin, we can detect some long-term trend in the particle fluxes. Because we have three experiments, the 97, 98, the 2007, the 2013, that were conducted more or less in the same station with the same methodology. So I was measuring an average for all of these separate experiments, and we have seen that for total mass fluxes, for example, the figures remain the same. So in time, there is almost no variation, since that the mechanism that supports particles toward the bottom, toward the center of the basin, still remain the same. And the situation is not the same as for regard of the biological particle fluxes, because they present a more different signal with a difference in time. But of course, this is just some little consideration, because the series is discontinuous and also it's very short. So the best is to continue and to have a longer series. So just trying to resume what we have seen is does sediment trap collect vertical or lateral flux? What we have seen that in the center of the basin, in the traps that is just below the photic zone, mostly collect the vertical fluxes from biogenical particle, whilst if we go in the deepest part of the basin, we have evidence also for lateral deflection with particles that are carried probably to wave-driven suspension or dense water cascading, and then a combination also of some particle coming from the upper side from the upper part of the water column. And if we move on the slope, the importance of lateral reflective material is even higher. This just to show you another example of what the time series of particle flux can be useful, because we don't collect also only particle, but also everything that fall out in the water column. And this is the use of the trap sample for another purposes, because, for example, during during 2004-2007, the presence of a plant of foraminifera, his name is Astigerina pelagica, that was never detected and was never presented in the literature of the Mediterranean Sea, was detected from some plankton net collection. And then we decided to check if in the trap sample collected in the period before that species was already present, and it was mostly absent. And then we checked also the trap sample that we have collected, so in the 2015 and 2016. And there, this is the white part of the bar is the presence of this Astigerina pelagica. And so we have evidence that the animal is present. So, summary, just to summarize everything, the open ocean convection is a controlling mechanism for the spring phytoplankton bloom and for the following fluxes. There is evidence of lateral action even in the center of the Southern Adriatic Basin, and there is a clear role of the dense water cascading in transferring particles toward the deepest part. The Barry Canyon system is an important role in the spreading and cascading of the Northern Adriatic dense water. We were able to define a better definition of the timing of arrival of dense self-water along the margin. The interaction, there is a strong interaction of the dense water cascading with the bottom morphology. The organic matter characterization could help to identify the particle provenance. We have identified some strategic sites for the long-term monitoring of this process. There is not a clear temporal evidence, but the time series is still too short. We have seen that particle fluxes can be useful to detect the invasion of some alien species, and also the zooplankton that is collected within the sediment house could be used for some wet food studies, as we presented from Emanuela in the next presentation. The effort of several projects and the collaboration with the many colleagues, and some of them are here.
Any question, or we can, okay.
Stefan, do you have maybe a seasonal variability of the fluxes of biogenic silica and carbonate in the traps?
Yes, we do.
Because at least maybe this, and what main feature do you?
So, mostly the organic carbon fluxes is a green one. Time, for example, these are results here. And this is expressed as a composition. Because usually organic concentration of organic carbon are higher when the organic carbon when the total mass flux is lower. But in case organic carbon is more important respect to opal.
So when do you have
this picture, for example, is not the right picture to the sketch to show you what you are asking. But we have higher value in the top trap respect to the bottom one. And the temporal variability is not so clear in terms of concentration. But if you look at the flux, you will get a picture of this surfaces that is very similar to the total mass flux.
Anyway, you have a higher concentration of biogenic silica in the upper.
Yes.
Right. And also carbonate.
Yes.
Okay.
Mm-hmm. Almost the double respect to the bottom.
Mm-hmm. Any other question? If not, yes. we can follow from Emanuele and then make a wrap-up.
Just quick.
Yes.
Maybe stupid, but because I don't know really well how the sediment trap works.
Yes.
But do you find usually litter or plastic items inside? And if you find them, what do you do with them? You just throw them away?
Yes. But the size of the trap is more or less 40 centimeters of diameter. And on the top of it, we have a screen that avoids the entrance of big fragments. We were discussing about looking for some particles from plastic.
It could be really interesting.
Yes, but one of the problems is that the amount of sample is often a little little. I don't know if it is enough, because we have samples that are only 2 or 10 milligrams for 1 pig, so it would be very difficult to find plastic in that concentration.
And the second one, the experiment was planned not for microplastic, or also for metal contamination determination. So sometimes we have problem if during the trial to use the same samples for different purposes. So, for example, cos'è che non abbiamo messo?
Zinc.
Zinc.
Zinc, because on the mooring line we have zinc anodes to prevent corrosion, and we found a lot of zinc within the traps. We were carrying them.
Okay,
Okay,
thank you.
Thank you very much.
Thank you.
Emanuela qual è? Questo,
Questo, no?
No, 11 dopo, questo delle 10.03. Facciamo 2.
Questo?
Sì,
Hanno lo stesso nome?
stesso nome?
Sì, ma non vuol dire che sono le due. Lo stesso nome. Ah, che bella.
Okay, I'm linking to the presentation of Stefano. This is a cooperative work with a group of CNR ISMA. That's correct. After the reorganization, and especially of Ilaria Conese, which was who was a PhD student, but now maybe she will be a researcher of this institute. And I'm a co-tutored. I was the co-tutor of a chapter of her thesis, and this is the work realized. Just very brief, because there are a lot of experts on zooplankton here, but this is for geophysics people, oceanographers. Okay, deep sea zooplankton, it's a key element of deep sea ecosystem, because we link the particular organic matter to higher trophic levels through both vertical migration, and it is also a prey of several megafaunal species, including the mercenary pelagic organism. So I just put this scheme as an explanation, very, very simple explanation of how the deep sea communities are supported by both vertical and advective flux, because apart some very local point of primary production characterized by chemosynthetic environments, the deep sea communities are characterized by dark environments, so no primary production occur. So mostly the food arrive from the surface, from the photic zone, by vertical flux. So the phytodetritus, which is composed maybe mostly by detritus, fecal pellets, so animal origin, and phytoplankton, dead phytoplankton cells, and of course they are remineralized also by the microbial loop, and these arrive to the I have a pointer, which is the central one. Okay. But I love to move.
Okay.
So it's okay.
You can do it.
That's my southern part. Okay. So the suprabenthos is also playing an important role in this sense. Then we have these swimmer flukes, according to Michel, 1984. So a group of specimens characterized by mostly mesozooplankton, but also macrozooplankton, mesopelagic fishes, for example, that perform vertical migration and transport organic matter from the photic zone to the deep sea community, supporting this megafauna, which is mostly composed by fishes, decapods, cephalopods, that are supported by this kind of flux. Then we have the advective flux, which is transport of organic matter from the, for example, canyon are funnel of transport of organic matter from the continental shelf to the deep sea. So we would like to identify which are the food sources for deep sea zooplankton communities in this work. And in this sense, we use this approach, which is the stable isotope analysis of nitrogen and carbon. It's a widely used technique for study trophic interaction in marine environments since the 80s, more or less. So it's not a recent technique, so it's just ancient in some sense, and it is recently applied to deep sea communities in the Mediterranean. We have a different study, mostly from the Western Mediterranean, Algerian Balearic Basin, another from the Eastern Mediterranean, but there were no data before this study from this area concerning animals. But yes, we have stabilized data for particles, of course. So the aims of this study is ambitious, somewhat. 3 aims: investigating food web structure of swimming fauna, that is macrozooplankton collected in the sediment traps that Stefano showed you before, to analyze possible seasonal patterns in isotopic signature and try to relate to the cascading events occurring in the area, and identify environmental drivers of the observed patterns, and in particular if cascading events have a role in such trends. So the study area, I skip this slide because Stefano described, and also not only Stefano described very well this area and the process of cascading, which is mostly of your competence. So I will skip this. You have already seen this data on the current speed and temperature on the bottom. We focus our work on these 3 mooring: FF, EE, and BB, as described by Stefano, and the BB is inside the Berry Canyon. The FF is on the adjacent slope, and the EE is the farthest one. So we expect it's the site less interested by the cascading concerning the incorporation of the signature in the body of the animal. Other environmental variables we consider are the chlorophyll A concentration from satellite data. These are for the 3 sites. For PP, the temporal data set is larger, so starting from March 2010. So we take also data 1, 2, 3 months before for chlorophyll A concentration, and the same for pore river discharge data. That's because we are from the bloom of phytoplankton as a of the input of organic matter on the surface and the arrival of organic matter on the deep sea bottoms. So generally in literature, this to be considered of chlorophyll concentrations until 3 months before. So November 2000 for EEA temporal data set. Maybe Stefano mentioned this. The caps are filled with formaldehyde, which has some effect on the stable isotope signature species, but we assume that the bias to our are processed, the animals were picked up and separated. That's the standard protocol. Animals are oven dried at 60 degrees. The isotope ratio, which gives even stable isotope and the common from C, the delta 15N, 15N and 40 nitrogen, and then we analyze the data by techniques, and also sample coupling, we use a mixing model to food source for the swimmer fauna. We are not familiar with this, the interpretation of these results, stable isotopes analysis, generally concern this of species within the food web. Consumer generally assimilates the heaviest isotopes, so the 15N, and excrete the lightest one, so the 14N. So usually we found an increase in this value, in the delta 15 nitrogen, through the food web. So the producer, for example, in this case, phytoplankton can be 1 per mil, the first consumer 4, 7, until the tuna in this case, which is the top predator that displays this kind of enrichment. So there is a fractionation because the theory is that you are what you eat plus something per 1,000, and this something is more or less 3 per 1,000. And concerning the carbon give us indication the source of carbon in the food web. So generally, from literature data, from a lot of experiments, meta-analysis, and so on, we know that very depleted carbon values are indicative of a terrestrial carbon source. We are around -29, -27 per 1,000. Here, intermediate value, already depleted value are indicated at pelagic source. So we are around -24, -21 per 1,000. And finally, an enriched carbon values, it's indicative of a benthic source. Okay. This is for the open sea environment. While if we want to, for example, discriminate between movement of a species, for example, in turtles, they are very commonly used for the movement of species from the inshore area versus offshore areas, we can also use this data as tracer of movement, for example. And in the case of carbon, the increase is slower, and is about 1,000 per mil at each trophic level. So generally, we obtain something like this by plotting the delta 13C data against delta 15 nitrogen data. So delta 15N indicator of trophic level, delta 13C what an organism eats, and we have a graph like this. Now we can also estimate the trophic level of a species based on this formula, which is very easy. Delta 15N of the species, we would like to examine it. This is a delta 15N of the primary consumer, that is our baseline. In the case of a pelagic food web, usually we use, and in the deep sea where we don't have primary consumer, we don't have herbivore species, of course, we can use a pond consumer, for example, or a filter feeder in general. And this is the trophic enrichment factor, as I told you before, it's about, from meta-analysis, around 3 per 1,000. So the results, thanks to collaboration also here with the OGS, Ilaria can identify 27 taxa. Not all the specimen were identified 2 species level, unfortunately, but for food web studies, the genus level is considerably more or less acceptable. So these are all the main group identified: Ketoniata, Cnidaria, 9 species of copepods or genus, Brachyuria larvae as a whole, they cannot identify species, Decapoda, Faucids, and so on. So first, the results, this is a cluster, the results of a cluster analysis. This analysis based on both tracer, delta 15 nitrogen and carbon, allow to identify 3 main groups. The first group characterized by filter feeders, Bibilias and Aperid, Acards and copepod, Cleopteropods, and a second group on the the extreme, which is characterized by species with the highest trophic level, and we found some copepods such as Calanus volandicus that from the literature is generally considered an omnivore species, but it seems in the deep sea to switch to carnivory. And in the middle, we have a lot of species that seems to, apart this one, which are unknown, with unknown feeding habits, because there are no data from literature, or because we don't have the level of species here, so we cannot determine exactly which is the trophic yield of the species. But there are a lot of carnivore, but also omnivore species. So it seems that in the deep sea communities, in these deep sea communities, species tend to be omnivore with a preference to carnivory, depending maybe from the food availability. That's a simple biplot, delta 13C against delta 15N, and the strength of the correlation between the 2 tracer somewhat indicate the array of food sources the deep sea zooplankton rely on, and specifically in period of no cascading, the strength of the correlation, which is quite low in general, is somewhat higher than in period of cascading, indicating that in period of cascading, maybe the array of food source is greater than in period of no cascading, when probably just a food vertical flux works for the deep sea communities. This is the results of a Kormanova very quickly. Both the factor cascading and site are significant, while the interaction term not, and the pairwise for site demonstrate that the two site BB and EE that are inside the canyon, and the farthest one differ from each other, while the two in the site in the canyon and on the adjacent slope are very similar in terms of isotopic signature. And the correlation with environmental variables were based on two types of models. The first one is a bivariate model based on the two tracers, data 13C and data 15N together, while the second one is based just as a univariate model, a generalized linear model, just based on the carbon signature. In the first case, we found that the best explanatory variables are chlorophyll A recorded simultaneously to sampling, and Paul River discharge recorded 2 and 3 months before sampling. However, the total variance explained by the model, according to the best solution found, it's not really high. And in the case of the GLM, the explained variance is a bit higher because we are working with just one tracer. And in this case, also again, river discharge 2 months and 3 months before are the best explanatory variables of the model, together with salinity. All the models, the type of correlation are positive, which is quite strange in the case we would have to consider cascading as a responsing variable, because it seems that increase in salinity provokate an increase in delta 13C, while we expect the opposite correlation in the case of the cascading effect. Okay, so the SIA results, that's a very simple Bayesian mixing model. We consider different food sources as input. In the case of the signature of the particular organic matter from the North Adriatic, we have to look at the literature because we don't have data from this area because it's out of the project. Maybe in the future we can do something different. So we take this data from the literature, also on the signature of the phytoplankton, while these two food sources, aged organic matter and soil organic matter, are from Leonardo publication of last year. And from this model, we can see that at the FF site, most contributor in terms of food sources to the deep zooplankton communities are the signature of POM from the North Adriatic. So we can see a sort of a kind of contribution of cascading of particle arriving from the North Adriatic to this community, but also phytodetritus. So we have the two inputs that more or less equally contribute as food source to the deep sea communities in this sense. In the second case, in the site inside the Berry Canyon, the signature, the food sources from the North Adriatic seem to be the higher contributor, together with phytodetritus, while in the farthest site, that is expected to be not influenced or less influenced by cascading in terms of food source contribution, actually is supported mostly by vertical flux, by phytodetritus. Sorry. Going to the conclusion concerning the first aim, food web structure, we depicted a complex food web with 3 trophic levels. That's a very important point, in my opinion, also for modelers, because when we try to fit the model, we need to be more detailed as possible, in my opinion. So from filter feeders, trophic level 2, to to carnivores, trophic level 4. Most of the species, as I previously said before, are omnivores that take advantage of particles that be or low particle phytodetritus or high trophic level origin, in the case of remains of organisms, of animals. According to the second hypothesis, second aim, the lack of significant differences in the interaction term cascading per site indicate somewhat effects of cascading parallel at all sites, although food webs seems to be different in each site, as we can see by the statistical differences between sites. The most explanatory variables in the correlation with abiotic data, environmental drivers, are chlorophyll concentration and river discharge recorded 2 and 3 months before. So this confirms that the signal between the input of organic matter from the surface or from advective flux are decoupled with what happened in the deep sea bottoms. And this is just a confirmation because other studies previously demonstrated that. And finally, considering the food sources, there is a wide spectrum of food sources for deep sea zooplankton originated by 2 different flukes. This is also a confirmation of previous studies. So we have a contribution more or less equal of the 2 input from plankton and indigenous material, but this is particularly true in the area which is influenced by cascading events. So cascading promote in somewhat the transport of indigenous material to these deep sea communities, while the site, for example, EE site, which is the farthest from the cascading events, seem to be not influenced by the events, or the communities there are not influenced by these events. And finally, just to go into the policy, why these studies are important. First of all, because the deep sea zooplankton, sorry for the error, but I added this slide last minute. It's a last minute slide. There is a general lack of data regarding zooplankton from all the sites, from micro, meso, particularly to mega. There are no data on mega zooplankton for the deep sea. That represents so a major gaps in the knowledge in terms of biodiversity and ecosystem functioning. We face now with the marine strategy framework directive, all the European countries, and studies like these. Also, we can take advantage of other sediment traps around the Mediterranean. In this sense, can be useful to feed. For example, food web models like ecopath with the ecosystem to better understand how these low trophic level compartments are divided. They are not represented by just 1 trophic level, but for example, in this case, we observe 3 trophic levels within these very, very small communities. Okay, so this could be useful also to identify appropriate indicators and criteria for the descriptor 4 or marine descriptor 4, sorry, of marine strategy, and the relative implementation to the deep sea. Because our current marine strategy is very beautiful. It's an ecosystem-based directive, so very ambitious. I think it's a very, very theoretically good approach, but lack completely of indicators and criteria specifically designed for the deep sea. That's all. In time.
Thanks, Manuela. Any question very interesting? More to learn plenty of things for us this time for the physical oceanographers. Yeah, Valentina.
Sì, ma also Michele.
Fai il bel
No,
like
just,
I didn't see you.
I'm happy that some of the identification made were useful. Can you explain me just a bit the plot that you made with the dot in black and white?
Yeah, they're very simple.
These one are.
These are all the stationed together because
the dot are samples, are collected.
no, are mean for species.
Each dot is a species or each dot is a sample?
Each dot is a species in cascading versus no cascading.
So you have different species here.
Yeah, we have different
Yes.
species, of course, because that's the usual, like this one. You have these.
Yeah.
We are here more or less. We don't have all the food web. When we plot all the data together, I did this exercise in another project when we sampled from the zooplankton, suprabenthos, benthos in the sea to top predators like deep sea sharks, and of course your range is different. You have the sharks here, but this is like a small picture of a part of the food webs. But usually, that's a paper. The results of a paper we are in second review in price oceanography. Hopefully, we will succeed briefly. And first, we separate the signature, cascading and non cascading at each site. But then one of the major concern of the reviewer was the interaction term of your analysis of the Permanova was not significant. So it means that the cascading effects exist in all the area. Okay, cascading is significant, site is significant, the interaction term not. That means that the event is apparently occur in all the areas. So you cannot do a separate analysis for each site. Although I'm not, at the beginning I separate the analysis. Yeah.
But, okay, you have a carousel of 12 samples, one for months.
Yeah, but we consider cascading merge, we put yes or not. Cascading, not cascading. Period of cascading versus period not cascading. So here you merge the individual from March to May, for example.
There is seasonality.
Yeah.
Okay. So cascading, not cascading. Seasonality and species all together.
Yeah, but we cannot. That's a problem. We don't have enough material to perform a seasonal analysis. We can do this just for some common species, and we did. We did this in the paper. I cannot show all the results, of course. That's a very.
But have you tried to separate the data from functional groups?
At the seasonal level?
Yeah.
We have also a graph in this sense. We have different data. I cannot show all the things. I prefer to show some, relating the 3 slides or results to the 3 aims. Yes, we have. Although we don't have enough material because the traps are not very big. There are different kinds of traps. So the material, maybe you observe the material. You don't have, of course, always the same species throughout the period. So, for example, when we analyze the seasonal variation, we have to limit only to the common species. Instead, you cannot discriminate the effect of different species occurring, so different feeding yield, different trophic strategies, and real actual seasonal variation. So it's not so easy. I did something like this when we did a work specifically aimed to look at seasonal variation in deep sea communities. But this is an opportunity work. The main focus of the work is not depicting the food web structure of deep sea swimmer and fauna collected in the sediment traps. So we cannot. We don't have enough material to do what you mean. And also we don't have the resolution we need to do something like this, because we can have several groups identify higher taxonomy level, general or brachyural larvae.
Yeah.
Inside brachyural larvae you can have different species.
The other question was if you try to compare the value imaging for a known species, imaging Italia-Messico, because it will recognize that between the 3 sites, because there are several examples of huge variations in the isotope for the same species, offshore. So
but they are both the 3 sites are offshore. The 3 sites are
have you
deep.
seen any differences between interest?
We have for some, I don't remember if Calamus helgolandicus is the case, but for some species we can compare side by side because we have, that's the problem I told you before. For some species, dominant species, more or less.
I give an example because I showed you the first one.
Yeah, what I saw also in my previous experience from the Algerian Balearic Basin, where we analyzed Calamus helgolandicus, is that the species in the deep sea seems to be carnivores in general, because the absence probably of fresh food source or different food sources may switch
The only
the.
speech of our biological. Last suggestion, compared to samples collected in open water. As you work on formaldehyde, you have a lot of samples, so you can compare.
But in terms of isotopic signature, if in the Adriatic, you have work, but from the surface,
Yes.
not from the deep sea.
Not deep sea, but just to see the difference that you find in the deep sea fauna. If there is a difference, it seems that there is a difference.
You have usually an increase of delta nitrogen and delta 13C with that. That's a common pattern found in literature by other work because of the remineralization and enrichment during the sinking of particles. So the POM is enriched throughout the water column. So it's usually expected that the species in the deep sea show higher values than the species on the surface, the same species, because of the microbial loop, remineralization, sinking pond, which is enriched by bacterial activity. So it's not so easy, in my opinion, to compare surface specimen with deep sea, because it's usual to find something like this.
The organism that you find in
This
the
is
trough
something that we can discuss outside.
are guys living in deep or not? This is the big question. If there are
More or
surface,
It's
someone that die and arrive just there.
No,
Otherwise, if they are
no, because you will find exuvia or something more mineralized. Because during the sinking, you cannot find entire animal. I work with, for example, with WP2 or Nanset net to collect zooplankton on the deep sea bottom up to 1,000 meters of depth in the Algerian basin, and you find entire animals. So they cannot sink from the surface. Of course, they live there. Deep sea zooplankton that live there, and we have different species. I don't know exactly the mechanism they do. I'm not sure they do vertical migration. I think there is a population of specimen that live there and not move because also of the stratification of water, difficulty to perform such kind of vertical migration. For example, among euphausids, we have meganictiphines that migrate, but we have nematoshellis which does not migrate. So.
At what time you have the train?
No, Michele, sorry.
There was just
I wanted to ask you, is there a relationship you told about the Po-river influence? By which way should occur? I mean, how do you discriminate that there is no contribution from other Apennine rivers and
from other
rivers from the Apennine?
Okay.
And how? I mean, most of the particulate could settle before, but is through the dense water transport or by which?
We suppose this because I mean the first hypothesis is to try to relate if this event, of course, they show previously as a response in the zooplankton community. Of course, we don't have all the elements to very, very perfectly reply to this question or to be fully sure of this. Of course, our model say this. In our model, the explanatory deviance is not really high. So probably there are other environmental variables that play an important role in this kind of studies in this web structure. Probably also live play an important role, because we saw from, and I discussed this with Leo, that leaf enters somewhat in the Berry Canyon, so probably there are other drivers that we didn't consider because we don't have all the elements to do this. For example, in the case of the food sources, for the model, I don't have any other source in terms of in literature, and we don't have data from other contributions. So the more data you have, the better will be your model, of course. But in our case, for example, we have to use literature data, which is from other year, for example, not the same year. That's a common error we have, but when you don't have all the elements you would need, it's better than anything.
Okay, thank you very much, Manuela. Thank you. Welcome. Now we resume for the coffee break. I will do it in 20 minutes just, and we have to resume here at 4 o'clock. Please be punctual there. Thank you very much.